cancers. So today I'm going to tell you about a new and critical role for the histone deacetylase sirtuin-6 in pancreatic cancer. So changes in chromatin dynamics are fundamental during cellular differentiation. So in ES cells, the chromatin is very hyperdynamic and globally accessible. And during normal differentiation, regions of the genome are silenced due to heterochromatin formation, thus allowing for only specific genes to be expressed, which determines cell state. So what I hope to show you today is that during pancreatic cancer progression, dysregulation of the epigenome endows cancer cells with the plasticity to override normal differentiation and reactivate early developmental programs. So chromatin modifying enzymes are one of the most frequently dysregulated class of proteins in cancer. As you can see from this recent review, many of these proteins exhibit either loss or gain of function during tumorigenesis. These chromatin enzymes modify specific chromatin marks. However, how their dysregulation leads to tumorigenesis remains poorly understood. So a large portion of our lab focuses on sirtuin-6 and its role in tumorigenesis. So sirtuin-6, or SIRT6, is a histone deacetylase that uses NAD as a cofactor to remove acetyl groups from the histone 3 lysine 9 and histone 3 lysine 56 to convert actively transcribed open chromatin into silent closed chromatin. So more specifically, our lab has shown that SIRT6 acts as a co-repressor of the transcription factors HIF and MYC by deacetylating H3K9 and K56 at the promoters of their target genes. When SIRT6 is lost, the chromatin remains hyperacetylated and thus more accessible for HIF and MYC to bind and increase the expression of their target genes. So I looked at the expression of SIRT6 in publicly available data sets, and I was struck by the fact that 60% of the pancreatic cancer cell lines exhibited copy number loss of SIRT6. So I looked at primary human PDAC, or pancreatic ductal adenocarcinoma tissue samples, and found that the vast majority had lost SIRT6 expression when compared to normal pancreas. So based on these findings, I wanted to determine how the loss of SIRT6 influences the onset and progression of PDAC in vivo. So I crossed our SIRT6 conditional knockout mice into an oncogenic, a very well-established mouse model pancreatic cancer, in which oncogenic KRAS is expressed specifically in the pancreas. As you can see, loss of SIRT6 cooperates with oncogenic KRAS to dramatically accelerate PDAC. The SIRT6 knockout mice had very highly disseminated disease. It was very aggressive and metastatic to both the liver and the lung. And this was true independent of p53 status, thus establishing that SIRT6 is a potent tumor suppressor in pancreatic cancer. I then wanted to determine whether SIRT6 controls the growth of established PDAC. So I made a series of murine PDAC cell lines from the tumors of our SIRT6 wild type and SIRT6 knockout tumors. And I found that the SIRT6 knockout cell lines had a greater propensity to form tumor spheres in low attachment conditions. I also found that the levels of H3K56 acetylation were much higher in the knockout, and this could be reduced by the re-expression of wild-type SIRT6, but not the catalytically inactive mutant. Restoration of wild-type SIRT6 led to suppression of cell growth in both 2D and 3D growth assays, as well as in vivo. I then was able to identify a series of human PDAC cell lines that exhibited either low levels of SIRT6 expression or high levels of SIRT6 expression when compared to the normal human pancreatic ductal epithelial cell line, or HPDE. I then re-expressed wild-type SIRT6 or the catalytically inactive mutant in some of these human cell lines and found that the wild-type, but not the mutant, was able to reduce the levels of H3K56 acetylation. This also led to a reduction in cell growth in both 2D and 3D growth assays. Conversely, knocking down SIRT6 in the normal HPDE cells led to elevated levels of H3K56 acetylation and an increase in cell growth. So this told us that SIRT6 can control the growth of PDAC in multiple contexts. So try and get at the mechanism by which SIRT6 is repressing the growth and progression of PDAC. We performed ChIP-seq for H3K56 acetylation and found that the gene that was most highly acetylated in the knockout cells was Lin28b. So strikingly, when I looked at the expression of Lin28b in my murine PDAC cell lines, I found that all of our SIRT6 knockout cell lines had high levels of Lin28b expression. So Lin28 is an RNA binding protein that directly inhibits the LET7 family of tumor suppressor microRNAs. It's highly expressed in ES cells and is necessary for maintaining the pluripotent state. In normal development, during differentiation, Lin28 levels are reduced and there's a reciprocal rise in the LET7 levels. So although Lin28 is normally silent and absent in normal healthy adult tissues, it can become reactivated in 15% of cancers analyzed and facilitates transformation, thus defining it as an oncofetal protein. So, however, Lin28 has never been studied in PDAC. So I measured the expression of Lin28b in our panel of human PDAC cell lines and found a beautiful inverse correlation between Lin28b expression and SIRT6 expression. When I re-expressed wild-type SIRT6, but not the catalytically inactive mutant, I was able to reduce the levels of Lin28b expression at both the RNA and the protein level in both human and murine cell lines. I also found high levels of Lin28b expression in our human primary tissue samples. This was in a large fraction. So try and get at how Lin28b, or whether it had any functional significance, I knocked down Lin28b in 3 SIRT6 low cell lines and in 3 SIRT6 high cell lines, and found that loss of Lin28b potently inhibited the cell growth in the SIRT6 low cell lines, but had no effect in the SIRT6 high cell lines. This was true in both 2D and 3D growth assays, seen here and here, and also in vivo, thus establishing that loss of SIRT6 defines a novel subset of pancreatic cancer that is highly addicted to Lin28b expression. To try and get at the mechanism by which SIRT6 is controlling the expression of Lin28b, we turned our attention to MYC. So Joshua Mendel's group has previously identified 2 MYC binding sites in the Lin28b promoter, and based on our previous findings that SIRT6 can act as a co-repressor of MYC, I performed CHIP for H3K56 and H3K9 acetylation. And as you can see, at the MYC binding sites, the chromatin is hyperacetylated in our SIRT6 knockout cells, suggesting that the chromatin is more accessible for MYC binding. When I knock down MYC in 3 independent cell lines, I'm able to suppress the expression of Lin28b, and this led to a decrease in cell growth in both 2D and 3D growth assays. So as I mentioned before, the main function of Lin28b is to inhibit the processing of the LET7 family of microRNAs. So accordingly, when we knock down Lin28b, we see an increase in all of the LET7 isoforms. We also see an increase in the LET7 target genes in our SIRT6 knockout mice. These are the insulin growth factor 2 binding protein 1 and 3. These are also oncofetal RNA binding proteins, and they were actually first discovered in pancreatic cancer and have been associated with poor prognosis, undifferentiated histology, and metastasis. So I measured the expression of the IGF-2BP1 in our panel of human PDAC cell lines and found that it correlated very nicely with Lin28b expression. I was also able to, when I re-expressed wild-type SIRT6, but not the catalytically inactive mutant, I was able to reduce the levels of IGF-2BP1, also when I knocked down MYC. So finally, in both the CCLE pancreatic cancer cell lines and patient samples, Lin28b high PDAC overexpress LET7 targets, and more specifically the oncofetal targets of LET7. And this is in multiple independently curated data sets. So this data allows us to propose the following model, that in normal healthy adult tissues, SIRT6 represses the expression of Lin28b by deacetylating H3K9 and K56 at the Lin28b promoter. Thus, SIRT6 acts as an epigenetic barrier to suppress Lin28b expression. When SIRT6 is lost in pancreatic cancer, the chromatin remains hyperacetylated and more accessible for MYC to bind and increase the expression of Lin28b. Lin28b can then go on to inhibit the processing of the LET7 family of tumor suppressor microRNAs, which leads to an increase in the LET7 oncogenic targets, such as the insulin growth factor 2 binding proteins, which ultimately leads in a more metastatic, aggressive, and undifferentiated PDAC. So I'd like to thank my advisor, Raul Moslavsky, and the members of the Moslavsky Lab, all of our collaborators here at MGH Cancer Center, especially Nabil Bardizi and Julian Fitamant for helping me with the immunohistochemistry. And I'd also like to thank our pathologist, Vikram Deshpande, our bioinformatician, Ken Ross, and Alan Gorn from MIT for performing the ChIP-seq. I'd also like to thank my funding sources. With that, are there any questions?
There's been an attempt recently to make very specific inhibitors of the HDACs, the isoenzyme. Is there a very specific inhibitor of HDAC6?
Of SIRT6? No, there isn't.
SIRT6.
SIRT6, no.
SIRT6.
No, there isn't. And nothing up to date. So,
But
we probably want to try and have activators. So Raul is actually looking for activators right now. So.
That's right, it's gone. You're right. But it would actually, an inhibitor ought to encourage the development of the tumor then, right? Yeah.
It could, yeah.
Yeah, in the ones that have it. Yeah.
I mean, a knockout is not always the same as inhibition.
It's true.
In a
It's
way.
True. Yeah.
So you don't know for sure what would happen if you were to bind it. I mean, you could end up stabilizing the complex.
It's true,
Have there
yeah.
been any even tool compounds?
No, nothing to date. But I think trying to inhibit Lin28b might be another really good way forward.
So, Susan, maybe you can answer this question. Have any of the HDAC inhibitors had negative consequences in terms of stimulating tumor growth?
I don't think there's any documentation of stimulating tumor growth, but lots of people talk about how you turn on both genes that are good for killing cells, but also you often turn on drug resistance genes. So that's more the issue. And if we could get a more specific model system, something like this, then you might see that. So the HDAC inhibitor is going to increase the acetylation, which is what you want here.
What percentage of human pancreatic tumors lack SIRT6?
So when I did the immunohistochemistry, we looked at 60 cores of PDAC here at MGH, and around 60% had lost.
Percent?
60 had lost SIRT6 expression.
It was a very good assay. You felt like it was
Yeah,
very
definitely,
sensitive.
very specific. Yeah, we had knockout to compare.
So recently, pancreatic cancers have been divided into several subtypes, glycolytic and lipogenic. Those correlate with prognosis as well. Is there any correlation between those that have lost SIRT6, whether they're glycolytic or lipogenic?
So that's actually an interesting question. So SIRT6 has also had quite a number of roles in controlling glycolysis. But when I looked in my model, I didn't see any indication that inhibiting glycolysis in any way affected the growth of the SIRT6 loss tumors. And I have not looked at whether it divides into either the glycolytic or the lipogenic phenotype.
So, not to play devil's advocate, so it would seem a little bit too linear. SIRT6, Lin28, LET, and cancer. And, but as you well know, you're affecting a lot more genes than that. Did you think of any? How did you get to Lin28? Or was that just a eureka moment? How many other things are changed? It seems a little bit, it's very nice work, but maybe a little too linear.
Yeah, so when we did the ChIP-seq, we found 184 genes that were specifically marked in the SIRT6 knockout cells, which was actually quite low in my thought for a histone deacetylase. And the Lin28 was actually the top candidate. So it was 30-fold higher in the SIRT6 knockout compared to the wild type. And looking down that list, there weren't a lot that were functionally relevant. So Lin28 really did come out as in this more, characterizing a more undifferentiated cancer. So yeah, I was surprised that it was the only one.
Now, at one moment you said LET7 was a tumor suppressor, and one moment you said it was pro-tumorigenic. So I don't know whether I misunderstood or it's both.
So it's sorry if I said that. So it's a tumor suppressive microRNA. So it controls oncogenic targets. So when it's lost, those oncogenic targets come up.
Okay. Loss of LET7. All right.
And then you found SIRT6 deficient, missing, low in 60% of PDACs.
That's right, yeah.
What else did you look at? Were you looking at levels of acetylation? And did you look at Lin28b in those situations, and what did you find?
Yeah, so I did not look at levels of acetylation. By immunohistochemistry, it's a little tough. But for Lin28, I found that it was elevated in 30% of those that were down for SIRT6. So it's only describing a part of the subset that loses SIRT6.